Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about operons. Now, an operon is essentially a bacteria's version of a gene sequence. So just a group of genes that's found in bacteria. Now, as humans, this may not seem super relevant to us, but in biology, everything is all intertwined, and so operons will help us understand the origins of our existence as eukaryotic organisms. So that's why we'll be studying operons in this video. First of all, we're just going to kind of go into the overall structure. Like I said, operons are found in bacteria, and they're groups of genes, and in bacteria, those groups of genes are grouped into units, and these units are operons. All of these operons are served by a single promoter and operator, which, like the promoter in the eukaryotic genes, will be what determines whether or not that gene is transcribed. And lastly, all the genes of an operon function together in the cell. So you may have a metabolic process, and there are 6 enzymes that need to be present in order for that process to occur. Well, then in bacteria, all 6 of those enzymes will be served in the same operon. All of them will be present together, and they'll all be transcribed together, and it'll all just happen at the same time. Because of this, it's actually a really streamlined process, having them in operons rather than individual genes. This allows, with only one transcription factor, many genes to be made that all coordinate. So that's kind of interesting for me. First, we're going to be talking about what's known as repressible operons. There are 2 types that we're going to get into. The first is repressible. So you can kind of see in this picture, I'm going to be going back and forth between it. First off, repressible operons will be transcribed unless a certain protein prevents it. So this is the operon as it normally is, and as you can see, there's nothing blocking the pathway. So this is the promoter, and RNA polymerase will just go right through, and it will just continuously transcribe those genes in the operon. This is actually known as the TRP operon, or it's named for tryptophan. So example is the TRP operon, which codes for enzymes used to make tryptophan. It actually makes the tryptophan from another precursor. We're not going to get into the actual process, but each of these 5 genes is essential in order to manufacture that tryptophan. Because it's a repressible operon, it can be turned off with the right conditions. In this case, it's tryptophan, and tryptophan will activate that repressor, which will then interact with this operator in order to stop RNA polymerase from just transcribing this operon. And so that's why it's repressible, because normally it will be transcribed, but it can be turned off, it can be repressed under certain conditions. Next we have inducible operons. And in the case of inducible operons, they will not be transcribed unless this certain protein is absent, this certain protein is absent. And the example we're always going to use is the LAC operon, or the operon that functions in a lactose breakdown. And so all 3 of these genes are going to be essential for lactose breakdown. And in this case, that repressor will always be present. It will always be blocking RNA polymerase unless lactose is present. And then it'll inactivate the repressor and allow RNA polymerase to come through. And so the reason for this is because if the cell has no supply of lactose, what is the purpose of creating those enzymes in order to break it down? And because it has that lactose present, that will be what causes the cell to break down lactose. That will be what signals the cell that it's okay to. In the case of the TRP operon and tryptophan, if it has an excess of tryptophan, it doesn't want to make more. So next we have positive gene regulation. If you notice, both of those forms, the repressible and inducible operons, were negative gene regulation, in which there's just like a negative feedback mechanism. And so with positive gene regulation, we want to promote the gene to being transcribed, and this will happen through these processes. There are various other proteins This is actually both in eukaryotes and prokaryotes, but again, we're still focusing on bacteria, prokaryotes, and that these will promote RNA polymerase its binding. This will be in that promoter, and when these other proteins are present, that will promote the gene transcription. That will increase the amount of gene being transcribed. The biggest example will be catabolite activator protein, or CAP. And what this will do is it'll stimulate the LEC operon whenever glucose is absent. Essentially, glucose is a much better supply of energy than lactose, and so if glucose is present, then the cell will not want to transcribe as much of the LEC operon. When glucose is absent, on the other hand, it'll be forced to use lactose, and thus CAP will stimulate the LAC operon, or the LEC operon. And so that's it for operons. They're really just as simple as that.